Good morning, I'm Andrew Holden from Auckland, New Zealand, and we've just had a fascinating session on the opening morning of Charing Cross this year, 2026, discussing paclitaxel and limus drug-coated balloons. We had some very important data on the SirPAD trial, and I'm honoured to be joined by Professor Stefano Barco from Zurich in Switzerland, and I'd like to congratulate you on the SirPAD trial being presented for the first time today in Europe, and also for the publication in the New England Journal of Medicine. So Stefano, this trial has a very unique design, quite different to many of the trials that we're used to seeing with drug-coated balloons, both above and below the knee. Perhaps you can start first by telling me a little bit of detail about the design of the SirPAD trial.
So the trial was designed as an academic, open-label, single-blinded trial that involved 2 trial sites and 44 referring centers in the whole Switzerland. The patients were referred from the referring centers for the intervention after outpatient screening. They were screened in the cath lab at 2 trial sites and then referred back for the clinical follow-up up to 1 year for the clinical endpoints and 5 years for the safety endpoints. We had an adjudication committee that was blinded for the treatment arm and indeed focused and adjudicated all suspected primary outcomes, so the composite of major adverse events, unplanned major amputation or target lesion revascularisation for critical limb ischemia, and all amputation events, including the secondary outcome. So it was really a broad adjudication of all possible primary and secondary clinical events. As I mentioned, the assessment of the clinical endpoints was limited to 1 year, and we didn't have any restrictions in terms of patient population. So no eligibility criteria based on PAD severity, target lesion length, or patient characteristics. The key eligibility criterion was actually based on the fact that patients were scheduled for an endovascular treatment for a symptomatic peripheral artery disease. And by doing this, we achieve a truly all-comers population with more than 90% of eligible patients being randomized in the trial.
And just briefly outline the exploratory secondary outcome that you found.
So in this hierarchical trial, we tested first for non-inferiority and superiority for the primary outcome. And then if superiority was met, we were allowed to test for superiority for a so-called key secondary outcome. That was the composite of any target lesion revascularization, so endovascular and surgical, critical and non-critical, or target limb amputation, either major or minor.
Perhaps now you can tell me the primary efficacy endpoint results and also the safety results of the trial.
So the primary efficacy endpoint occurred in 8.8% of patients in the sirolimus group, as opposed to 15% of patients in the control group. And what we observed was a similar and consistent direction in terms of risk reduction for the 2 components of the primary efficacy outcome: unplanned major amputation and target lesion revascularization for critical limb ischemia. So this is important in order to emphasize that the main results were not driven by TLR alone. We are studying, and we studied the primary safety outcome, that is all-cause death, at 1 year for the primary analysis and at 5 years for the secondary analysis. And in the primary analysis, we have shown that there was no difference in terms of all-cause mortality at 1 year between the 2 groups. So now would be interesting to see the 2-year data at the end of the year and the 5-year data in order to reassure also the agencies that there is no increased risk of death with this device.
I guess there's going to be some comments and criticisms of any trial that this is a very unique, I think, endpoint design. Can you make some comments? Typical trials focus more on patency differences between the 2 treatment arms. Perhaps you could explain your rationale there.
So the point is, we come from the venous world. So we are to some extent naive in this field. And what we appreciate is that in all other vascular fields, the primary endpoints that is based just on surrogate markers, binary stenosis, primary patency, need for TLR, are not accepted and cannot be used to drive recommendation in the guidelines. So we were asking ourselves, why is there no trial that is focusing on our clinical endpoints in peripheral artery disease? So if we focus on claudican patients, the main criticism to the, let's say, available trials is that they use a scale, the Rutherford scale, that is hardly to be reproduced in an open label setting. So it's very difficult to interpret these results. B, there was no improvement in quality of life in functional scales. And C, all the trials have been performed in an era where we didn't have dual pathway inhibition optimization of the lipid-lowering therapies. So in this setting, we wanted to confirm that there is an improvement in terms of our clinical endpoints, including TLR. If you move to the below the knee setting, then we know that the drug-coated balloons to date failed even for the surrogate outcomes of primary patency, might increase the risk of distal embolization of death. And in this setting, it was quite a bet to design such a trial that really aimed to cover the all-comers population. But by doing that, we had two tools in order to allow us to also make statements concerning the subgroups. The first tool was the sequential randomization. So we had patients that had been randomized for the first 50% of the sample size, and after 600 patients, we were allowed to do a predefined interim analysis with sample size recalculation in order to see whether we have enough power to achieve our goal.
Yes.
This was the first tool. And the second was the hierarchical analysis that we performed. So we wanted first to test for non-inferiority for these hard clinical outcomes, in order to be allowed to then perform an analysis for superiority for the individual outcomes.
Yes.
And these, I think, were the two ways that paved the way to success. And the third one could be considered the fact that we really had very sick population with almost 50% of patients with either acute or chronic critical limb ischemia, which is unprecedented in any PAD trial.
Yeah.
And if you don't have the events, then you cannot prove superiority of any device over any other device in terms of clinical outcomes.
So as we try and interpret this data into our current clinical practice, we've learned here from Serpad that we've obviously reached a superiority with statistical significance for a sirolimus drug-coated balloon, the Concept Medical MagicTouch balloon over standard angioplasty. I guess a lot of practice around the world now is with other drug-coated balloons, primarily paclitaxel drug-coated balloons. How do you think we interpret this data in the background of having paclitaxel balloons? And do you think we need more trial evidence to really compare those technologies?
So I cannot make any formal recommendation, of course, and I would not suggest using one balloon over another. This is simply for the guidelines. We produce the evidence, or the best available evidence, to support the use of this type of balloon, MagicTouch sirolimus-coated balloon, for an all-comers population of patients, moving from patients with shorter lesion and femoropopliteal disease to very sick patients with a rate of a major amputation of 30%. Across the spectrum of patients, we could prove that there is a clinical benefit. The second question would be whether you would prefer a sirolimus-coated balloon over a paclitaxel-coated balloon. Well, I cannot answer this, but the data that we've seen today concerning femoropopliteal patients with treated with either balloon was actually very positive, and I'm talking now of the Sirona trial. So if you just focus on primary patency, there is also in that perspective reassurance that sirolimus-coated products are effective and safe.